All right, so what you might want to do here, you can open up the PowerPoint and mindlessly stare at the PowerPoint, like some of you kind of do on a daily basis, or you can open up the homework that's due tomorrow and kind of fill out the homework as we're going, because what I'm going to talk about is very much going to align with the questions you have in the homework. So, did it say due today? Oh, I know it's due tomorrow. My bad. So if you haven't done, you might want to have that homework open so you can kind of match up to see how you're doing. I thought I had it posted for tomorrow. Sorry. So it's up to you. And if you don't have it done, you can smile because you can open it up and finish it right now as we're going. So we're going into DNA. We're going into double helix. The first part is kind of historical context. We start with talking about how we came to understand that DNA is the molecule of inheritance. We know far more about genetics than any of these scientists did, at least the early ones. Again, we said it a few times. Mendel had no idea what chromosomes were. Mendel had no idea what DNA was. DNA is this kind of inert molecule. Doesn't really react to anything. Didn't really think it did much. I didn't really think it played much, had much of a role with living organisms. We obviously know that it does. All right, so we're going to go through kind of the history of how we came about to understand that DNA is the molecule of inheritance. We're going to walk through a few scientists. Overall, the tests so far, they're pretty good. They're okay.
Really?
I think so. On the multiple choice part. The written part's up and down, but the written parts, they seem tricky, but they really were pretty easy if you kind of understood the question.
Did everyone take the answer test?
No, you can't, because I still have at least one or two, I think, you can take. Yeah. All right, so off that, let's talk about Frederick Griffin. Frederick Griffin was a scientist, did work back around 1928, and he was studying pneumonia. He was studying bacterial pneumonia, so pneumonia that's caused by bacteria, and he was studying how pneumonia affects mice, affects mammals. So that's what he was looking at. In his studies of pneumonia, he really noticed that there were two key types of pneumonia, two key strains of the pneumonia bacteria. There was an R strain. Now, R stands for kind of a rough. The R strain had kind of more of a rough outer coating. You can actually tell the difference by looking at a microscope. And then he had a smooth strain that we denote or show with an S. It had more of a smooth outer coating. That rough outer coating produced toxins. Right? So produced toxins actually were harmful to the organism. I'm sorry, the rough that did not produce toxins. My bad. So the rough did not produce toxins. The rough was harmless to the organism. The smooth strain, however, were slightly genetically different, and they produced some toxins that made them pathogenic, harmful to the mouse, would make the mouse really sick and possibly even die. Right? So he was kind of looking at this. He was looking at different types of bacteria, and he started kind of getting into his experiments. His first step was realizing that the R strain, when injected into the mouse, would really have no effect. The S strain, when injected into the mouse, would actually cause the mouse to oftentimes die. So this is kind of what his experiment looked like. So here is the smooth strain, which is the pathogenic strain. Right? When injected into the mouse died. Here is the rough strain. When injected into the mouse lived. Now, to kind of break down the experiment just a little bit more, he then heat killed the bacteria. So he took a test tube with the smooth bacteria in there, and that's my artistic ability showing fire, a Bunsen burner. He heated up the bacteria. So he killed it. He basically caused the bacteria, all the proteins to break down, to denature. The bacteria was no longer living. When he injected this into the mouse, as we can see here, the mouse was happy to begin with. Right, whiskers, all that good stuff. But we injected this in there, took the needle. It's a mouse.
Sounds like a camel.
No, it's a mouse. He injected it into the mouse. The mouse is fine. No big deal. The mouse was not harmed. It was heat killed. All right. So now he's got an interesting problem here. He said, all right, so hey, we inject the rough. No big deal because it's not pathogenic, so the mouse is fine. We inject the smooth. All right. Mouse dies. We inject the heat-killed smooth. Right. The mouse is fine. So now we're going to understand, hey, guess what? There's something going on here. So now he goes to really the next aspect of his experiment. And what he does, he gets another beaker, another test tube. And this one has rough in it. And the rough is living. And what he takes some of the heat-killed smooth. So he takes some of that heat-killed smooth bacteria and puts it into the same test tube with the living rough bacteria. We let them culture for a little time. Let them kind of grow together. And then he injected it into the mouse. That's a syringe, by the way, Kenny, in case you're wondering. And what happened to the mouse this time? Mouse died. The mouse is sad. All right, the mouse is dead. So the mouse died. Now, we had no idea what controlled inheritance at this point. No idea. And we still don't know. He has no idea that it's DNA. But he's starting to understand that there's not this kind of weird life force. It's not like this Jedi magic trick that controls inheritance. What he started to understand is, well, guess what? There's got to be some molecule in these cells, because you don't need that to be a living cell to kind of transmit this information. That's what he realized. He realized that, these smooth bacteria that are dead, right, they're not kind of giving this weird force to the rough bacteria, they're dead. There's got to be some molecule that is being passed from the smooth bacteria to the rough bacteria. And that's really what he came up with. He came up with, hey, guess what? There's some molecule of inheritance here. And he came up with the process of, oh, it's on the board, I was looking for it. He came up with the process known as transformation. Simply saying that material from one organism, we know that material is what? DNA from one organism can transform or change the genetic makeup of another organism. So Griffith was really the first one to come up with transformation. Again, he had no idea what the molecule was. We know it's DNA. If you had to guess, what do you think his molecule was? Not RNA, although RNA is a better guess, but not RNA. Proteins. Why proteins? Why does everybody around here, as soon as we start realizing there's some molecule that controls inheritance, why does the entire scientific community guess proteins?
It's like there are 20 possible protein amino acids for combinations, and also like it's proteins are compounded part of like essential processes.
Right. When you look at an organism, you look at the chemical reactions of an organism, what's controlling all those chemical reactions? Proteins. When you're looking at mechanisms, right, what's controlling movement across membranes? Proteins, right? Everything in our bodies are controlled by proteins, so it made sense that, hey, guess what? The molecule of inheritance is probably a protein. They're wrong, but that was their best guess. Okay, so Frederick Griffith got us to the point that say, hey, guess what? We now have some evidence to say there is some molecule, some inert molecule, some non-living factor, right, that carries genetic information, heredity. That takes us to Oswald Avery. And this is what we know. We just kind of stated it. The world knows that some molecule carries genetic information, right? We don't know what that molecule is. You can go back to the macromolecules, right? Those are probably the main guesses everyone's going to have. Either DNA, RNA, right? Nucleic acids, carbohydrates, lipids, proteins. Those are the guesses. Again, with everybody really leaning on the protein one. Oswald Avery really set out to try to really determine which one it actually was. He had a really smart experiment. It's really just Griffith's experiment with kind of an extra little twist, as it says right here. So this is Oswald Avery's experiment. I'll give you a moment to kind of read through it, because you kind of see the methods of his experiment up here. I know it comes to like, I hear it every day, and you get that boost of energy, like, oh man, it's so awesome that we have AP Bio last period. I just need that one little boost of energy to get me to the end of the day. It's great that it's, this amazing AP Bio class instead of. All right. So this is the experiment here, right? Anything that ends in AAC is a what? Is an enzyme. And RNase is an enzyme that probably does what to RNA? Right? Degrades it, right? So RNase degrades RNA, protease degrades proteins, DNase degrades DNA, lipases degrade lipids, and carboase, right? It degrades carbohydrates. It could be amylase, could be a number of different ones there. So as we look through, right, we have our heat-killed, right? Our heat-killed smooth bacteria. I inject that into a mouse. Mouse is going to be fine. I take that heat-killed smooth and I start to culture it with my R strain, my non-pathogenic strain. But before I do that, I mix it with some enzymes. So in this test tube here, all of the RNA molecules are degraded. So there is no RNA, functional RNA presence. We culture for a period of time. We take that, the co-culture together, we inject it into the mouse, and what happens to the mouse? The mouse dies, right? Why does the mouse die? Because RNA is not the main molecule of inheritance, right? So we get a mix of the smooth and the rough strains, right? We get a conversion there, transformation does occur, right? And our mouse dies if we inject it. Here, we do the same thing with proteases. Everything's functional except the proteins, right? We take that culture with the protease, we mix it with the rough, right? And again, we take this culture and we inject it into the mouse. What happens to the mouse? Mouse dies. Why? Proteins aren't the molecule of inheritance. All right. Take the same heat-killed S strain. We mix it with a little bit of DNase. Okay. Now we culture it with our rough. Are any of those rough bacteria transformed into smooth bacteria? No, because DNA is degraded. DNA is the molecule of inheritance. So therefore, if the DNA is degraded, transformation does not occur. We inject this into the mouse, and the mouse lives. Yay, happy mice. Right? We do the same thing with lipase. Again, the mouse is going to die because lipids are not the molecule of inheritance. We do this with carboase. It's kind of a little made-up one right there. Carboase, we mix it. The mouse dies as well. So the mouse dies in every instance. Mouse gets sick in every instance. Or we could just analyze the sample to see if any of the rough was transformed into smooth, which is being highlighted here. And in every instance, transformation did occur except for the situation with the DNase, which is evidence that DNA is the molecule of inheritance. Now again, we know that. They didn't know that back then. And DNA was really kind of this unknown molecule. It doesn't do much. DNA doesn't react. It's not very obvious in our bodies, right? It's just kind of there. So it didn't really have any function up to this point. So why did we think it carried the most important function there is of holding heredity? So that's that. Everyone okay? So we've now progressed. We know there's a molecule of inheritance. We know that that molecule of inheritance is DNA. Except nobody believed them. Right? People were still stuck on proteins. Right? Good scientists are skeptical. They repeat experiments and they try to adapt and vary experiments to prove results. So now we come to Hershey and Chase, the Hershey and Chase experiment. And they are really looking to do the Avery experiment again. They cut out the carbohydrates, they cut out the lipids, right? And they're really just focused on DNA and proteins. Okay? So they're just doing the exact same experiment again. They're testing the same hypothesis, but they have a different method in this process. And they use these little moon lander things. This is actually an electron scanning microscope. Those are actual bacteriophages. They look like moon landers, little legs, right, and the head part. So they use these bacteriophages, these viruses. A couple things about viruses. Are viruses living?
No.
What's the main piece of evidence we use against viruses to say that they are not living? What's that?
They don't perform metabolic processes.
They can do some things. What's the main thing they can't do on their own? They can't reproduce on their own. So there's some, they do carry out some chemical reactions. So it depends on how you define metabolic process. But they can't carry out some. The one thing they can't do on their own, they can't reproduce on their own. Living things have to be able to reproduce on their own. So they're not really considered living. How bacteria, especially bacteriophages, how they reproduce is by hijacking the mechanism of a host cell. So what they do is they take their genetic information, which you can kind of see right here. We know it's DNA. It's injected into a bacterial cell. In this case, they really target E. coli cells. And they force the E. coli to make more of the virus, make more of the bacteriophages. They hijack the components of their host cell, which is a great experiment. It's a great experiment because, for the most part, there are some other components, but for the most part, viruses are made of, they're very simple, proteins and DNA. Nice and easy. So now we have the two culprits, protein and DNA, and we have an organism, or not really an organism, a virus that really just contains the two. So we have the setup of a great experiment here. And that's what Hershey and Chase did. They looked at the T2 phage, which is just the T2 bacteriophage, and they looked how it attacked E. coli. So what did they do? Again, I'll read through this, but I'm going to walk you through the image as well. They grew one back to T2 phage in the presence of radioactive sulfur. That radioactive sulfur targets proteins, right? So therefore it's built into the protein molecule, so it's radioactive, so we can see it. They grew another batch with radioactive phosphorus, right? Why? Because phosphorus is a component of DNA, but not really a component of proteins. So now I have two different samples, one radioactive proteins, one radioactive DNA, right? And then we culture them with E. coli. I'm going to go through this again, but I'm going to use the image to kind of talk through it. You got that?
Okay, why does,
Question.
Why does sulfur work?
I think it's cysteine. I forget which amino acid, but there's a main amino acid in the variable group, in the R group. There's sulfur atoms there, and that's where the sulfur molecule ends up. The reason why they use sulfur is because sulfur is not present anywhere in a DNA molecule. So we know it's only going to be targeted on proteins. Yeah. I think it's cysteine. We can look it up, but it's in the R group of one of them. All right. So here's our experiment. Here's our Erlenmeyer flask, right? That's being cultured with bacteria and viruses, right? We're making a whole bunch of these bacteriophages. And this one is cultured with radioactive sulfur. So as the proteins are being built, as the components of the viruses are being built, those protein components are being incorporated with radioactive sulfur. So all the protein here is radioactive, and we can see that nice and neat because we see our bacteriophage. The pink of the bacteriophage is representing the protein coat on the outside, and the pink is representing that protein coat is now radioactive. Here's our second batch down the bottom. We're just making a whole bunch of viruses. We have a culture of bacteriophages, right? It's in a radioactive phosphorus medium. So as DNA molecules are being made, right, that radioactive phosphorus is being incorporated into the DNA. And we can see it's showing highlighted here. The radioactive material is blue, which is the DNA component, which is inside the protein coat. So here we can see the protein coat, radioactive sulfur. Here we see the DNA molecule labeled with radioactivity. All right, now what we need to do is we need to take these viruses out and we need to culture them with E. coli, right? And we just culture them for a period of time. Once we do that, we really just need to isolate them. So, they run them through a blender real quick just to kind of, break up the materials, to separate the materials a little bit, and then we centrifuge it. When you centrifuge something, what goes down to the bottom? largest, densest material. So if I were to centrifuge just like one of our cells, right, you would most likely get kind of the mitochondria, right? The larger organelles would be down at the bottom of the material, of the filtrate. And then the smaller ones, the ribosomes, would still be kind of floating around at the top. So here, the larger components are bacteria. So when we centrifuge this system, right, you're going to get the bacteria down at the bottom, and you're going to get the viral component up at the top. All the viruses will be left behind because they're smaller. So when we centrifuge this setup, and we analyze it for radioactivity, we just simply look for radioactivity, we notice that the radioactivity is in the fluid above the pellet. This pellet is where the bacteria are concentrated. That's telling you that the protein, the radioactive protein, never entered the bacterial host. It stayed separate from the bacterial host in the viral components. That's why this pellet is not being measured as radioactive. In the second system, right, where we have the radioactive DNA, we look for radioactivity. We see that the radioactivity is located in the pellet, which is telling you that that DNA, the radioactive DNA, has now entered the host, has entered the bacteria, and therefore that is the evidence that DNA is the molecule of inheritance. DNA is the transforming factor, right? The protein can't transform it from the outside. So by looking at this, we see that DNA is the molecule that actually entered our bacteria. Therefore, this is just supporting Avery's experiment from before, saying that DNA is the molecule of inheritance. Question?
Would it be wrong if you say like RNA is also a molecule?
No, you're not wrong. It definitely holds genetic information. In fact, you have a lot of viruses that are based off of the RNA molecule, retroviruses. And could you, when you're doing these experiments, could you see some noise with some RNA molecules playing a role in this? Absolutely. But the RNA molecule isn't set up to be replicated and repeated. So whatever level of RNA you have initially is the level of RNA you're going to maintain. You understand? So therefore, the DNA molecule could be incorporated and continually be expressed. So it can continue to make more and more and more RNA to elevate the levels. Whatever RNA molecule comes over is kind of, you're at that level, so you're only going to go down from there in terms of expression. Does that make sense at all? Yeah. But could you absolutely pick up some RNA noise in some of this? Yeah, you could. Yeah, because it's part of it. Okay. So DNA is the molecule of inheritance, which is backed up. We're finally able to say that. Now we know that DNA is the molecule of inheritance. Now we want to know, how does DNA work? What does DNA look like? Right? How does it hold our genetic information? Now we know DNA is double helix, right? The rungs of the ladder are the nucleotides, but it took a little while to get there. And one of the key experiments in this process was done by a guy named Chargaff. He came up with, anyone in my class before? Blank rule.
Base pairs.
Came up with base pairing. He came up, Chargaff. If I come up with a rule, I'm going to call it Angus's rule, right? So he called it Chargaff's rule. So he came up with Chargaff's rule. Chargaff's rule is real simple, right? Adenines kind of pair up with thymines, and cytosines kind of pair up with guanines. That's it. We already know it, right? But he came up with it. And again, he didn't know the structure. He didn't see this, but you can chemically analyze a substance for levels of certain molecules. So I can come up with a test to analyze, how many adenine molecules are present there, and that's what he did. He looked for concentrations. And if you realize he had a DNA sample that was 40% adenine, when he tested it for thymine, it came up to be about 40%, right? And therefore, you have the last 20% was broken up between cytosine and guanine. So it doesn't get any more complex than that. Any type of question you could see with this would simply be saying, if I gave you cytosine equals 15%, if cytosine equals 15%, what percent of the substance is going to be guanine, what percent is going to be adenine, what percent is going to be thymine? This is going to be 15. That equals 30, right, which leaves us 70 left, so it's going to be what? Yeah, 35%, 35%. That's the most difficult question you'll ever have with Charcot. It's that simple. So we already know base pairing, right? A's come up match up with T's, and C's match up with G's. And that was Charcot, which was key. It was key to kind of understanding how they kind of complement each other and how this kind of complementary base pairing occurred. Who's the next scientist in the progression here? Who's the one that got screwed? Rosalind Franklin. Right, so Rosalind Franklin worked with a partner, Maurice Wilkins, and their famous image, Image 51. There's a lot of controversy around this picture. It was thought that Watson Crick, Watson Crick were the father of the DNA molecule. They really kind of came up with the DNA molecule. But it was thought that Watson Crick stole this image out of Rosalind Franklin's lab. There was a lot of espionage going on. It's not quite that clear. They were all doing work in London. I think it was London. Pretty sure it was London. But it was definitely in England. I'm assuming London. And they were working, she was working at King's College. Watson and Crick were working right next door. There was a lot of communication. They knew each other. And they didn't quite get along. So it's not necessarily that they didn't know each other. They snuck in and stole Rosalind Franklin's data. They were kind of supposed to kind of work together, and they had some budding personalities where, Rosalind Franklin she was very strong in her opinions. I'm not saying that in a bad way whatsoever, but she was very forward, liked to have conversations and debates and liked to argue with people to kind of come to an understanding, where her partner, Maurice Wilkins, was a little bit more laid back. It didn't necessarily like any type of conflict. So she was supposed to work with Wilkins. They didn't quite get along. Wilkins kind of got along with Francis Crick, right? So they kind of got along a little bit. So the image just got passed around. Watson Crick ended up seeing the image. The image 51 was key to them really coming up and figuring out that the double helix is the structure of DNA. But it's just a lot kind of going on at that time. I'll talk more about Watson Crick in a second. But why was this image so important? And what did she do? So Rosalind Franklin and Maurice Wilkins, she really specialized in a process known as X-ray diffraction. All it really is that. Right now, photons are coming out of the projector. They're hitting my hands. Some are bouncing off. Some are passing by. And you see a nice shadow of my hand behind. That's all X-ray diffraction is, except they're not using a light source. They're using radiation. They're using like the radiation you get from an X-ray. Okay? And they would shoot it at a crystallized molecule. She was looking at a whole bunch of different molecules, just like this. So here's your X-ray machine. We get a focused beam of X-ray, of radiation. It hits that crystallized molecule, and it creates an image of that molecule behind, basically a shadow image of that molecule. You can see this image isn't very clear, right? And this is image 51. Now it doesn't look like much. And it doesn't really look that important unless you know what you're looking at. And if you do this, you start to see why that image is so important. You start to see it's really the crossroads of the double helix. Here's our DNA molecule, a very simply drawn DNA molecule. This is our double helix, two strands kind of winding around one another, right? And this is what you're seeing is the interaction of the two. So it really was an important image. Not too many people, it's debated whether she knew what she even had. She wasn't completely focused on DNA, so nobody really knows if she knew that it could be the image to the double helix. But when Watson and Crick saw it, they knew what it was. It was that last piece of information. They've been building DNA molecules for a long time, and they finally came to the understanding of, hey, guess what? It's a double helix structure. And they developed it from there. That's just if you want to look at DNA replication, you can go there, but it's kind of simple. All right, so there's James Watson and Francis Crick. Yes, James Watson is a racist. He's really fallen out of the good graces of the scientific community. He went as far as saying that I don't believe I don't believe that I'm racist, but I definitely don't think that all races share the same level of intelligence. Yeah, he said that. And then he said, ask any employer that employs people of other races. Yeah, so he said that back in 2007. Since then, he's really, doesn't really get work that often. And he actually is trying to sell. He's tried to sell his Nobel Prize. In fact, I don't know if he even sold it. This is back in 2007, 2008. So he tried to sell his Nobel Prize. Francis Crick, he's a good guy. He's still fine. They're not together. They just did this work together. Francis Crick, there's the Crick Institute over in England. He still does a lot of good work. I mean, he's older now. He's like in his 80s. But he's done a lot of good work and everything. So just kind of crazy with them building it. This is the actual structure they built. And the key thing, not only was the key thing of seeing the structure from that X-ray diffraction, photon 51, but from that photo, you can start to see the interactions between molecules. You can actually do measurements to get a measurement of distances between atoms, which allow them to get the proper measurements between the atoms in their structure. And James Watson wasn't so much of thinking he was better than other people, not only other races, but also women as well. He wasn't exactly in favor of women in the laboratory. He would have actually learnt about all those measurements, and he actually would have learnt about photo 51 15 months earlier when he sat in on a lecture by Rosalind Franklin, that she gave all the information he used to actually build this molecule. But because he sat there and didn't listen to a single thing, he got nothing out of it and had to end up, luckily with the photo ending up back in his lap. So kind of crazy. All set?
Oh, I just have one question.
Yep.
Not exactly sure what the question is.
Dominant recessive, links, unlinks, independent assortment, X-links, those are inheritance patterns.
Okay.
So which one does that display? Okay, so yeah, kind of crazy. Not all scientists are good people. So this is our molecule of inheritance. Here's our DNA molecule. Couple things. You can kind of see the A's match up with T's. The A's are double ring structures. The adenines are double ring structures. Where the thymines are single ring structures. You can see double rings match up with single rings. Helps to keep even spacing. If you have two double rings match up, the molecule would start bending and twisting a little bit too much. So that's why A's match up with T's. You can see why guanines don't match up with thymines, because guanines have the ability to form 3 hydrogen bonds, where thymines and adenines only want to form 2 hydrogen bonds. So you can see the ring size, double ring, single ring, and the amount of hydrogen bonds they want to form, which is really what's forcing the complementary base pairing. It's why adenines match up with thymines and not the C's or the G's. You can see the double helix here. So the double helix, the kind of twisting helix, where the molecules are parallel, anti-parallel, which I'll mention in a second. And then the key components here. So here are the nitrogenous bases in the middle, right? A's, T's, C's, and G's. This is where your genetic information is held. Here's your sugar phosphate backbone. So the phosphates and the sugars. If you remember a nucleotide way back when, 5-carbon sugar, phosphate group, nitrogenous base. The phosphate group and the sugar make up the backbone, which leads to the strength of the molecule. It's why we find, fairly intact DNA in the Neanderthal bones, because the molecule is rigid. It does not really degrade. It holds its structure very well, which is smart because it holds our genetic information. We don't want it bending and breaking all the time. So just kind of talking about parallel, I'm going to mention this again tomorrow. We start going into DNA replication. Not only is the molecule parallel, it's actually facing in opposite directions. So it's really considered anti-parallel. This would be a parallel setup. This would be an anti-parallel setup. And we note those directions by 5' and 3' ends. Nothing big about 5', 3', just how we note it. On the 5' end, you have a phosphate group. On the 3' end, you have a hydroxyl group. The reason why this is called the 5' end is because we named the carbons. Organic chemistry has a naming system. So we name the carbons. That's carbon 1, 2, 3, 4, and that carbon coming off the chain is carbon 5. So the phosphate group is attached to the 5' carbon. Hence, we call that the 5' end. You don't need to know that, but I'm just telling you why 5', 3'. You need to know 5', 3', where the hydroxyl is attached to the 3' end. And one last thing, and we're done. Purines and pyrimidines, right? We just kind of talked about how the double ring structures always match up with the single ring structures. You do need to know purines, pyrimidines. It's really easy. You mess it up all the time. Pyrimidine, the second letter is a Y. 2 of the DNA bases have Y in their name, right? So pyrimidine, cytosine, and thymine both have Y's. They are pyrimidines, right? Guanine and adenine do not have Y's. They are purines. That's how you remember the purine pyrimidine breakdown.